Hi everyone, today we are going to talk about the experiment of measuring lactase enzymatic activity. But before we start, let's get a quick review of the background information of enzymes. Enzymes are macromolecules in living cells. They are abundant in plants, animals, and bacteria. Enzymes function as speed boosters of reactions. A reaction happens when free energy can achieve the activation energy required. Enzymes lower the required activation energy, which saves the time elevating the free energy and makes reaction easier to happen. In this experiment, we have a major goal, measuring lactase catalytic effectiveness by observation and data statistics. First question, why do we choose lactase as our subject? Noting that the catalytic efficiency varies between different enzymes, we should only focus on one kind of enzyme when measuring. But why is lactase rather than other enzymes? To answer that, we should focus on 2 points, relevant to human disease and commercially available. It is estimated that nearly 90% of Chinese people have lactase deficiency in their intestine, which hinders the process of hydrolyzing lactose into glucose and galactose. Bacteria in the intestine instead decompose lactose into lactic acid, hydrogen, methane, and carbon dioxide. These products may trigger intestinal spasm, diarrhea, and flatulence. Therefore, getting the data of lactase catalytic efficiency would benefit the pharmaceutical process. However, we aren't going to use lactose as the substrate of lactase in this experiment. Instead, we use OMPG. This is because when adding lactase to lactose solution, we will find out that the solution remains colorless. This is not helpful for observation and further analysis of efficiency by measuring light absorbance. If we use OMPG as a substrate, the solution will be color developing. This could happen because OMPG mimics lactose shape, enabling it to bind to the active site of the lactase. In this experiment, we need 14 centrifuge tubes in total. 2 are for control groups, one without adding lactase and the other without adding the substrate OMPG. Then separating experiment group into 4 small groups, varying in incubation duration, respectively 30 seconds, 60 seconds, 90 seconds, and 120 seconds. Each group contains 3 repetitive experiments. P.S. Don't forget to add a stop solution immediately after the incubation to stop the catalytic reaction. By qualitative analysis, no color developing of the control group shows that without enzyme or substrate, the catalysis will not occur. In experiment group, we can observe that the depth of color increases with the incubation duration. To make quantitative analysis, we can pipette the solution in all the centrifuge tubes into different holes on 96 well plates and put it into microplate reader to measure the solution's light absorbance in each hole. Then, if we draw the absorbance incubation time graph by using the figure of average absorbance of solutions in each incubation duration, we can find that the line is approximately a straight line. This inspires us that the effectiveness of lactase is constant for every second of the incubation time. The mismatch on the 90-second point may be due to experimental error. Recall that the major goal of this experiment is to find out the effectiveness of lactase, which means we need to find out OMPG's amount of conversion per second per microliter of lactase during the reaction. The graph we have got shown in the results section is the absorbance incubation time graph, which does not directly reflect the amount of substrate conversion. Therefore, we need a standard curve to map the absorbance to the concentration of the solution. However, we don't have this kind of standard solution to draw the absorbance concentration graph. So I would just briefly discuss about how to calculate if we have a standard curve. First, since absorbance is in direct proportion to the incubation duration, we can choose an absorbance value at any incubation duration we have and correspond to the standard curve to find out the concentration of the solution at any absorbance value. With solution's concentration of each incubation duration in hand, we can multiply the volume of the solution to get the total amount. Using the amount to be divided by the incubation duration, we can get the conversion of OMPG per second. It hasn't finished yet. We should further calculate the conversion of OMPG under a unit of lactase. Thus, this value should be divided by the amount of lactase. In this way, we have reached our goal by qualitatively and quantitatively measuring the catalytic efficiency of lactase.